Yes, we are live.
We are live, yes. Okay, সকলোলৈকে নমস্কাৰ আৰু আজি আমাৰ এনাজৰীৰ প্লেটফৰ্মৰ জৰিয়তে ক্লাইমেট ডায়লগছ ৱে ফৰৱাৰ্ডৰ আজিৰ সেশনটো আৰম্ভ কৰিবলৈ লৈছো আৰু আজিৰ সেশনটোত আমি পেলিঅ ষ্টাডিৰ বিষয়ে কথা পাতিম ডক্টৰ সুপ্ৰিয় চক্ৰৱৰ্তীৰ সৈতে আৰু welcome, sir, to our session. Thank you so much for being here. And before going to the session, let me give a short intro about you, and then we will start the discussion. Dr. Supriyo Chakraborty is from West Bengal and completed his schooling in Kharagpur. Then he pursued a career in geoscience, graduated from IIT Kharagpur, and then earned his PhD at a Physical Research Laboratory, or PRL, at Ahmedabad in 1995. There he studied about past climates using natural records. After his post-doctoral work at the University of California, Santa Barbara, and San Diego, he returned to India, worked at several institutes, and then joined Indian Institute of Tropical Meteorology, IITM Pune, in 2007. And there he headed the Mass Spectrometry Group to advance research on ancient climates and initiated the MetFlux India Project. There he also taught us 2 classes for us on paleo study, and that's when Vidyut and I came to know about that branch of climate science and we learned a lot about him. And this project measures, his MetFlux project measures carbon fluxes to understand how much carbon dioxide forests absorb and release. In collaboration with Tezpur University, Dr. Supriyo also sets up a meteorological tower at Kaziranga National Park in Assam. And that study found that Kaziranga forest might release more carbon dioxide than it absorbs, which sounds very interesting. And Dr. Supriyo's research focuses on monsoon reconstruction using the natural archives, isotope hydrology, moisture dynamical processes, greenhouse gas dynamics in urban and natural environments, quantification of ecosystem greenhouse gas fluxes, and energy transfer processes at natural ecosystems. He has about 100 peer-reviewed research papers to his credit, and he is also the coordinating lead author and an editor of the book Assessment of Climate Change over the Indian Region, a report of the Ministry of Earth Sciences under Government of India that was published in 2020. He is also the recipient of the Certificate of Merit Award by the Ministry of Earth Sciences, Government of India. Dr. Supriyo Chakraborty is currently a visiting faculty at the Department of Atmospheric and Space Sciences at Savitribai Phule University, which was earlier known as Pune University. And beyond research, he is a nature enthusiast. He promotes conservation and sustainability, enjoys cycling, and writes poems about natural processes for outreach. And while we were circulating the flyer, I also shared a Facebook post where he has shared a poem written on Northeast India, and that was such a beautiful poem. So if you get some time, and please visit that and please give your comment. Okay, welcome, sir, to our discussion, and we are honored to have you here. It's a long time since our PhD days, and we also look forward to learn from you a lot, like we used to do in our past classes during our PhD. Yeah, welcome back, sir. So before going to discussion, do you want to share some thoughts with us?
Yeah, thank you for introduction. And this is really very good opportunity for me to disseminate particularly the kind of work I have done, especially in the context of Northeast India, and that is also very specifically Kaziranga National Park. So I will try to give you an overview of the kind of work that we have done. It is mostly, of course, it is not the kind of work I have done in Northeast India. It is strictly speaking not paleoclimatic work, but it is mostly modern work. But there is a thread between the modern work and the paleoclimatic work. That is what the isotope, how we use this isotope, basically this isotopic techniques, that also I like to elaborate in the course of time. So I think it is mostly based on discussions, right? So if it is so as per your questions and queries, I can answer and respond to your queries.
Yeah, okay, thank you, sir. Actually, while with the flyer, I was going through your poems, the all poems that was shared in your Facebook profile, and I took 2-3 lines from that, from one of the poems. I forgot. I think it was about ocean and coral reefs or something like that. And the 2 lines was like. Let me read out for you.
That oxygen 16, 17, 18, right?
Yes.
Sorry, carbon 13, 14, 15. Yeah.
Yes. It was
Sorry,
like
not 15. Carbon 12, 13, 14.
Yes, and paved a way for scientific analysis to perform carbon dating. And that time, yes, I was thinking of again requesting, as I said in my Facebook post, that I was thought of requesting you to write a poem on Northeast India, and which is surprisingly, anyway, we received. So let's start from there, sir. So
Sure.
In that 2 particular line, so you are telling about 3 isotopes of carbon. And there you mentioned like 3 isotopes of carbon, and the way that it can be a way to scientific analysis to perform carbon dating. So let's start from there. What is the carbon dating, and which branch of climate science it's connected to, and what is it?
Yeah, that question actually is very pertinent, because carbon dating is a very useful tool in not only in climate science, but in archaeology and oceanography and many other things. So just I just like to try to explain these things. You know that carbon has 2 stable isotopes. One is carbon 12, and that is the natural carbon. That means it has 6 proton and 6 neutron. And then it has one isotope, which is having 1 extra neutron. So that gives rise to a mass of 13. So carbon 12 and carbon 13, these are 2 stable isotopes. And their abundance is very small, roughly 1%. So out of every 100 carbon atom, you can have only 1 carbon 13. So this much of abundance. Now, apart from these, carbon also has a radioisotope, and whose mass is 14. So that's why you call it C14 or carbon 14. But this radioisotope, firstly, its abundance is extremely small. Unlike 13, which is 1:100, 1:100, and carbon 14 is 1:10^12. That means for every 10^12C, you can have only 1 14C. And so this much very small abundance. And carbon 14, since it is radioactive, so it has it has a life cycle. So it is born in the upper atmosphere because of high energy cosmic rays that enters our Earth's atmosphere in the upper portion of the atmosphere. So these cosmic ray excuse me, contains a lot of nitrogen, proton, nitrogen, and various kind of high energetic particles. So they interact with atmospheric nitrogen, and as a result, carbon 14C is formed. So this is called an NP reaction. So one neutron is because of 1 neutron, 1 proton is ejected, and then 14N it is converted to 14C. So once it is converted to 14C, it quickly becomes 14CO and then 14CO2. Eventually, it is oxidized to carbon dioxide. So this carbon molecule is 14CO2. And one of the beauty of this carbon molecule is that it gets quickly distributed in all the sphere, that is atmosphere, and from atmosphere it goes to ocean, that is hydrosphere, and then it also goes to biosphere, that is plants, and also it goes to lithosphere, that is soil, but that time scale is longer. So generally it quickly gets distributed first in the atmosphere, then in the ocean, and then in the plant. So they are very quick process. So every living organism, they contain small portion of 14C. Even we also as a human being or any animal will have 14C because they feed on plants. And so they inherit this 14C. So everybody has 14C. And the idea is, as long as an organism is living, it is continuously interacting with the atmosphere. And because of this interaction, so any living material is constantly getting 14C in its body. But at the same time, 14C is also getting decayed since it is radioactive. So it decays continuously. The half-life is about 5730 years. So on a global time scale, the number of 14C produced, I mean at the upper atmosphere, there is no other production of 14C except at the upper atmosphere. So the number of production of 14C is approximately equal to number of decay of 14C per unit time. So this is on a global scale. Locally, it may vary, but on a global scale, this maintains a more or less equilibrium. So as long as this equilibrium is maintained, you can say that in any body, supposing in science, as we use a box model, supposing if we consider a box kind of thing, and there you have production, so that is called input, and then you have destruction, or in case of decay, so that is output. So in general, this input and output are same, so that there is no excess buildup of 14C or there is no deficiency of 14C. So when this equilibrium is maintained, and now suppose the organism dies. So at the moment it dies, this interaction will stop. So there is no interaction, so it is not interacting with the atmosphere. That means an organism, it is not eating anything. So 14C intake is stopped, but decay will continue. The moment it happens, you can say the clock starts. So this is you can say this is the clock starts. That means when the organism dies, or scientifically speaking, when it gets dissociated from the atmosphere, where from it gets its 14C source. The source of 14C basically is the atmosphere. So the clock starts and it will decay exponentially. So as a result, what will happen after certain time, for example, after about 5730 years, the number of 14C atom, whatever it had accumulated, that will become half. So we can say it has passed approximately 5730 years. So now, if we supposing we do not know if we want to the date or how long it has been it was disconnected from the atmosphere, for that we have to collect the sample and we have to find how many 14C is remaining. So that we can do certain instruments. So these are called like scintillation counter, or present day it is used by accelerator mass spectrometry. So they will tell you how many 14C is remaining. So if we know that number, then we can get their date. So this way, for example, we can date any archaeological sample, we can date any breeding sample, we can date any carbonate sample. We can date also water mass. For example, if we want to know the past ocean circulation, then we can also get the carbon 14 signature from water. Now, water itself does not have carbon, but ocean water has a lot of carbon dissociated in this. which is called dissolved inorganic carbon. So a lot of carbon is there. So if we collect 1 liter of water, we'll get this DIC, the significant amount of DIC, that is dissolved inorganic carbon. So if we do carbon dating of this water, so then we'll get its age. Actually, how age, the concept of age is slightly different. So the concept is, as long as the water is in the surface, means surface sea, so it is continuously interacting with the atmosphere. So as a result, it is also gaining 14C, some 14C from the atmosphere. But when this water mass, supposing you know that in ocean there is upwelling and downwelling of water. Now because of downwelling process, this water goes down, and then when it goes down, it gets disconnected from the atmosphere. So in that process, what will happen? It will no longer incorporate 14C, but at the same time 14C will continue to decay. So how long it has been disconnected from the atmosphere, that we can calculate if we do the radiocarbon analysis. So this is a very good tool to know about the past ocean circulation. So this way, and when we do the carbon 14 dating, also you have to do carbon 13 analysis, that is 12C 13C ratio also you have to measure. So you measure both 14C 12C ratio as well as you measure 13C 12C ratio. So and 13C 12C ratio, which is called delta 13C. So this will give you other information, for example, about the productivity. So in ocean, how much, what is the productivity, means when productivity is high, that is phytoplankton activity is high or phytoplankton activity is low, that we can assess by getting the delta 13C value. So this way, if we get both this information, that is delta 13C and delta 14C, so we can tell about natural processes. So that is why the meaning of this line is C12, C13, 14. If we measure these things, we get to know about the natural processes.
Yeah. Yes, sir. So, and this branch of climate science where you study this, that is called paleo study.
Yes.
Right. So, so if we have to summarize, so that is the paleo study where we study like past climate, and then we compare 14C, the two isotopes of carbon, 14C and then 13C.
Yeah, actually it is not compare, but we get two independent, actually there are two parameters. They are connected, very much connected, but there are two parameters and they give you different information.
Yes.
So for example, suppose in atmosphere, you are tracking a air parcel. So you can measure its temperature, you can measure its relative or Q value, that is specific humidity. You can have some liquid water content, etc. Similarly, in ocean water, so you are measuring two parameters. One is its stable isotopic values, another one is radioisotopic values. So when you combine these two parameters, you can understand or you can study processes. Processes in this case, for example, ocean circulation, you can study. So this is the idea.
Yeah. So yeah, and if I have to ask you, so then why do we need to do the paleo study? So
yes.
So
yeah.
in
Why
one,
do we?
let's say in one line.
Yeah. Okay. Why do we need paleo study? That is simply because in the current context, we see that how Earth is warming, right?
Yeah.
And it is warming very rapidly. Now the question is, did our Earth have similar warming in the past? Actually, it had. So now from the past, we can get some information about what may happen in the future. So this is one aspect. So how this warming this warming took place in the past, and whether the current warming is similar to that kind of changes or it is something different. So that we can assess. This is one thing. And then second thing, Earth has since we know that Earth also experience several ice ages. Generally, they come every 100,000 years. So the current warming, actually, current warming, it may lead to ice age also. There is a very simple explanation why the warming may lead to ice age. For example, we know that in the North Atlantic, a lot of this heavy, I mean, ocean water, it sinks. Because of strong evaporation, the surface water gets heavy, so they sink. And then the conveyor belt starts. So now the sinking process will continue if you have a very oil mixed water in the North Atlantic. But now supposing because of heavy melting of polar ice, so they will cover the ocean surface, and polar ice generally they don't have much salinity. They are almost very free of salinity. So you have low salinity water, and underlying you have high salinity water. Now, so it will stratification will take place. So because of the stratification, that low salinity and high salinity water, the mixing will be weak, very, very weak. And the moment mixing becomes weak, the conveyor belt will be highly affected. And if the conveyor belt, in fact, the past climate or the paleoclimate, it showed us that during the glaciation time, the conveyor belt was very weak. If the conveyor belt becomes weak, then we are very likely to have another ice age. So this way we can take lesson from the past, and then we can learn what may happen into the future, so that we can take some mitigative action.
Okay, let me simplify this part. So you said in paleo study we studied the past, and then let's say for particular different times for different era, we study the pattern, or we study like the time of that weather or climate of for that time, and why
Yes.
did it happen, right? Let's say we find the characteristics in simple words for that time, and then we study now whatever the observations we are receiving now or projecting, and then we are studying whether we are going to have the similar characteristics in certain times from now, and then projecting those characteristics to the past climate, and then accordingly we are projecting whether we are leading towards the ice age or a very warmer phase, right? Am I right, sir?
Right.
Yes.
That's right.
Yes.
Basically,
Okay.
we take lessons from the past.
We take lessons from the past. Yes.
Yes.
Yeah.
So
I'm
I
just
can
trying to. Yes.
Yeah, maybe I can give you another example. See, about 3 to 5 million year ago, the atmospheric CO2 concentration was very similar to today's.
Yes.
You know, about it exceeded 450, something like this. And now the current CO2 concentration is 420. But that time, this is called Eocene time. So this Eocene time, the temperature was very high, was much higher than what we have today. So now the question is, if we get this CO2 concentration, maybe another 10-20 years, we are likely to reach 450. So if we reach 450, what will happen? The Earth's temperature will be same what we had during the Eocene time. So these are the questions we can investigate, and we can get information about the past climate, and then we can apply what may happen in the future.
Right.
That knowledge we can apply.
Okay.
Yeah.
Yes,
I wanted to like ask something like the title of today's session or discussion. We had put Northeast India, a ticket to time travel. So from what you explained, I understand that you explained the part of time travel, how we can go back in time and check climate in different eras and see the characteristics, what Nabhanita was saying. But now, how come Northeast India is a ticket to time travel. Can you put things in the context of
Yes,
Northeast India?
yes.
That how do we study past climate in Northeast India?
Right, right. That is a very pertinent questions, and why did I choose this thing? So that will be clear. I mean, I like to tell you one of my experience there, that as I told you, I did not do so-called paleoclimatic work in Northeast India, but very few with some collaborators I did, that is in some lake sediments. But what we have done, see that where we have tower, so near this tower we collected sediment samples, so-called vertical profile. That is, we dug about 1 meter borehole, three boreholes we dug, and then we collected for roughly about every centimeter, or maybe not every maybe every 5 centimeters. So approximately 20 samples we collected vertically. Now
That's from Kaziranga.
this way, that is from Kaziranga. Yes, it is in the Kaziranga. Now this Kaziranga, so this we did isotopic analysis, as I told you, the 13C and 14C both. Now 14C gives you the date, and 13C will give you how the stable carbon isotope varied with time. Now when we did this dating, we found something very peculiar. Peculiar in the sense, what we found is that if we take the first few samples, so we got up to age about 5,000 years. Zero means, supposing, see, you know that natural sedimentation process, it is a natural process, and it continuously deposits. For example, a river is dumping sediments into the ocean, so it is a continuous process. So you can imagine that sediment is slowly it is building. So one sediment layer is falling into another sediment layer. Then after some time another sediment layer is falling. So you can understand this is a continuous sequential process, and one after another layer are forming. Now if you do dating, so you are expected to get the date is continuously increasing with depth. The top one is the younger and the lower ones are the older. So this is what you are supposed to get. But now in Kaziranga, what we found is that up to about 5,000 year we got age, 0 to 5,000. 0 is modern value, and 5,000 year is up to 5,000 years. But after that, means when you do the next sample, do you know how much we got? We got something about 18 to 19,000. So 5,000, instead of getting 7, 8, 9, 10, 11, 12, you jump to something about 18 to 19. Now, why it is so, that we do not know. But there, what possibilities means, so-called it is not published, but because we do not have sufficient sample. Actually, in order to have confidence in these things, you should have at least 10 borehole you have to dug and collect so many samples and see whether you are reproducing the same results in most of the cases. So that we could not do because of logistics, etc., funding, etc. But at least in 2 boreholes, we got this kind of information that after 5,000, it is jumping to nearly 18 to 19,000. So what does it mean? It means that during this deposition test, for example, let us consider this deposition was taking place from 30,000 to 25,000 to 20,000, then 20,000s, then it all the way it's coming to 5,000. So that means roughly about from 5,000 to about 15,000, let's say 20,000. So 15,000 layer is missing.
Okay.
So how it is happening? Whether it is, firstly you have to check whether it is right, means you are getting the same signal in other boreholes, and secondly, if it is so, how it is possible. So this way, I mean, we have some hypothesis. The hypothesis that most probably during, and you know that last glacial maximum took place, it is about 20,000 years ago. So this is a very important phase in Earth's history, because that time huge ice sheets were formed, particularly in northern hemisphere. India, it was not so intense, but entire North America was covered by very thick ice sheet. Entire North America. And now, and sea surface temperature in the equatorial region was down by about 2.5 degrees centigrade, which is huge. You know, SST reducing by 2.5 globally, it is a huge signal. So this is in a nutshell, this is the condition in during the LGM, so-called LGM, that is last glacial maximum. So now, so most probably this continuous sedimentation took place up to about LGM. LGM peaked something about 21,000 years ago. But after that, something happened. So it may what it may have happened that the Brahmaputra River, probably it had a different course before this, what we have today. So that time probably it had a different course. So this is a very, very interesting study, and if it is true, then it will be really very good. Means probably means we can if we can give a proper explanation, so that will have a lot of implications about the environmental consequence, what happened in this region.
Yes.
Yeah.
So
yeah, I think we just received a question from one participant, and I was also thinking about that. So basically, sir, this was from your Kaziranga experiment, right? And
Yes.
that was used to study what did you study? Isotope analysis. That's what you said, isn't it?
Yes. Yes.
Yes. And you had some past studies on Zero Valley,
Zero Valley, yes.
right?
Zero
And
Lake, actually. Yeah, Zero Valley Lake, yeah.
yes. And did you study some that Meghalaya caves as well, ever?
Okay. That is actually, I did not study from Meghalaya cave, but actually we studied from, there is a cave called, there is cave in Andhra Pradesh that is called 𝗏𝗏𝗃. So 𝗏𝗏𝗃, we studied that things, but I am aware of this other people, some one German group, they did very extensive analysis of Meghalaya cave.
Okay.
And
So,
tell me, huh?
So, sir, this question, like what kind of information can we gather from speleotherms from the caves in Arunachal and Meghalaya? And my question was also in the same direction, that your study from the Kaziranga, that isotope analysis, you state a very significant information you probably might discover. And also, what kind of information you discovered from that study on Zero
Valley Lake.
And
Zero Valley
yes,
Lake.
Zero Valley
Yeah.
Lake, and then Meghalaya caves. What kind of study or information we achieve from those?
See, Zero Valley Lake, actually, I basically, I got involved with some of my collaborators. They are from also Birbal Sun Institute and Botany Department in Calcutta University. So, so they did pollen analysis. You know that many trees release pollens, and these pollens actually, some pollens thrive in particular climatic condition, like some of them prefer humid condition, some of them prefer maybe arid condition, some of them in slightly higher temperature condition, like this. So if you know the characteristics of the pollen, so then by doing this pollen analysis, you can tell at least qualitatively what kind of climate you had that time. And my contribution is basically in this case is doing the radiocarbon analysis to tell you that depth, at what depth particular the sediments. Actually, what we generally what we do is we try to collect a core sample. You have to drill a core in the lake bed, whether it is lake or it is ocean. Generally, what we do, we drill a core. So then it will give you a cylindrical shaped core, and then we have to do the dating of this core. So once you do the carbon dating, so it will give you the how long climatic record you can expect from this core. Supposing a core is about 1 meter, or 2 meter, whatever, and it is giving, let us say, 10,000 years. Means top and bottom. If you do radiocarbon dating, it is giving, let us say, 10,000 years. So then what you do, you collect, you take several layers, and from these layers you pick up, you can pick up pollens, you can pick up other carbonate bearing materials like gastropods or cells, like there are any freshwater cells are also there. So these freshwater cells, if we do the isotopic analysis of these freshwater cells, that will give you one information. What kind of information you give? I can explain you very simple. See, supposing it can give you out the lake level, when the lake level was higher and when the lake level was lower. For example, if it rains very heavily, you can expect the lake level was high. But supposing, but I am not telling about internal timescales. These may be happening in several decadal timescales or maybe century timescales, because internal timescales we cannot resolve. In paleoclimatic record is very difficult. Every year what is happening, that is very difficult to tell. But you can say let us say decadal timescales. Few decades, let us say, it rained very heavily and relatively heavily, and few decades it rained deficient rainfall years. So what will happen during the deficient rainfall years? The lake level goes down, evaporation is high because it is dry condition. So during the dry condition, what happens? Because of very intense evaporation, from the water, see, water like carbon, water, in water you have two isotopes of oxygen, like 16O and 18O. Similarly, you have hydrogen and deuterium. So when water vapor is produced, that is, water evaporates, the lighter isotopes, they escape faster than heavier isotope. This is very, very simple physics, because lighter isotopes, they need less energy compared to molecules which have heavier isotopes. So H2's 1H16O will escape faster than 1H18O. So as a result, what happens when lake level goes down because of intense evaporation? So surface water, it is called isotopically enriched. That means 1816, very simple speaking, 1816 ratio will be more in case of evaporation compared to a normal condition. So a particular carbonate material like a gastropod cell, if it is born during this time, so it will inherit the isotopic signature of lake water. So that time, what will expect? You will expect the delta 𝐸 is going higher. So in deficient years, delta 𝐸 value will be higher, and in excess rainfall year, delta 𝐸 values will be lower. Okay. So if you do the isotopic analysis, then you can tell about the lake level. That is what we did in this case.
Yeah. Sir, I have a follow-up question here. So yeah, I'm again simplifying a little bit, that we studied the past climate, we found the characteristics, we studied do the isotopic analysis, and yeah. And then now my question is, when we do the paleo study, we always study about the climate, like how was the past climate and what could be the future climate with that related to that, right? So like my exact question, I don't know how to put that. Like, what could be the different branches? Let's say we studied a pollen, and we study pollen proxies. That's what this is, right? Then we study the isotopes, and then we find out about the vegetation, like how was the vegetation in that past climate, what was the characteristics, and
right.
So these are all, like, I'm to ask, like, about the paleoclimatic studies, or these are like different branches of paleo study. Let's say yesterday, I was talking to someone, and then I just mentioned about that we have Meghalaya has so many caves, and then there are like so many studies are going on. Then the person commented quickly that, oh no, this is more related to geoscience. So how do we differentiate? Then when we study, when we see something, a research, how do we understand that this study is telling about that, okay, this is a climate science, telling about climate science, or this is about geoscience, or yeah.
Actually, yeah, this is a good question. I mean, especially in the point of view, for example, the young people who want to pursue a career in climate science or paleo science. So in that respect, the question has a relevance. You know, so actually, paleoclimate is basically an interdisciplinary science. Truly speaking, it is an interdisciplinary science. Most of the people you see, they of course they come with geology background. Why geology background? Because the isotopic techniques they are taught in geology. Generally, they are not taught in physics or chemistry or mathematics. They don't get exposed to what is isotopic technique, etc. So that is one reason the geology background people come here. But when they get involved in these things, and then secondly, another thing what you need to know is that some geological processes. For example, how the deposits take place. For example, how a speleothem is formed. How does it form? So you have to have some idea about the processes, and that is purely geoscience, purely belongs to geoscience. Formation of speleothems, formation of any other, for example, corals. So they also get slowly deposited in marine environment. So these are purely geological processes. So you have to know. And actually, probably in school you have learned that there are three kind of rocks. One is igneous rock, one is sedimentary rock, another one is metamorphic rock. So every school child knows this thing. So, but what is important here is that the most of the record, climatic record, they generally we can get this record only from the sedimentary rock, not from other two kind of rocks. Igneous rock, I mean, it is very easy to understand that igneous rock cannot have any fossils. And secondly, they don't have layerings because they don't deposit sequentially. Igneous rock, how it happens? Because the hot magma, it comes from the Earth's interior, and then the moment it comes to the surface, it cools. And because of this cooling process, the rocks are formed. So there is no question of sequential deposition. So, and metamorphic rock, it is no, I mean, these things probably, that either igneous rock or sedimentary rock can be metamorphosed. It means if they are subjected to very high temperature and pressure, they get transformed into metamorphic rock. Basically, it is a transformed rock. So similarly, it also does not have any layering. So as far as climatic studies is concerned, igneous rock and metamorphic rock has no value. So that geoscience part is gone. Now, if you have some idea about this sedimentary geology, so this is a branch called sedimentary geology, then only you can undertake a paleoclimatic or climatic studies. You can get climatic information from these things. So up to this, you need knowledge of geology. That is how the layers are formed, whether fossils are there or not. So those things. But now when you do the isotopic analysis and many other things, so then you need a lot of knowledge in particular in physics. This probably you have heard a person called 𝗬𝗪 who got Nobel Prize in chemistry, but he is the pioneer in the isotopic systematics. First time he proposed that if we do the isotopic analysis of these carbonate samples, then we can get temperature information. How it is possible? Actually, isotopic studies, one important thing is that isotopes, basically the isotopic ratio, they are controlled by vibrational frequency of the molecules. There are different modes. One molecule can undergo vibrational oscillation or rotational. It can have vibrational motion, rotational motion, and it can have other kind of motions. So only when it vibrates, so then this depending on the bond strength, the bond will be broken depending on the frequency of vibration. And you can theoretically calculate what would be the 18:16 ratio. That is purely thermodynamics. So thermodynamics will tell you, for example, we very often use one particular technique that is called isotopic exchange. So isotopic exchange, what does it mean? Supposing if you keep water and carbon dioxide, let us say carbon dioxide, it is in gaseous phase, and water, it is in liquid phase. And that you have kept in a small vial, let us say. So it is half filled with water, and the overhead space, you have carbon dioxide. Now, if you leave it like this, then supposing you maintain a constant temperature, then naturally what happens, the heavier isotope of oxygen in water, it will go to CO2 phase. And the lighter isotope of oxygen from CO2, it will go to carbon dioxide liquid phase. So this is called isotopic exchange reaction. This is a purely physical process. This is not a chemical process, because water remains water, carbon dioxide remains carbon dioxide. There is no change, but isotopes get exchanged. So how many isotopes will go from and what controls this exchange? Basically, this thermodynamics. If they do exchange, then the system will gain the minimum energy. That is the criteria which will drive this exchange process. And how many this exchange takes place, that you can calculate using thermodynamics. So you need a lot of physics knowledge. Of course, we do not do these things. But if you want to know the mechanism, then you have to do these thermodynamical calculations. So this way, you need both geological knowledge as well as physical knowledge. And then, as you know, now in order to analyze the data, you have to do a lot of statistical analysis. So you have to have some good knowledge about mathematics, particularly statistics. So this way, it becomes an interdisciplinary science.
Yeah. So, sir, like moving from geoscience to climate science, the paleo study, if the study has to become a climate science related study, then the main point we need to keep in mind that it should involve the sedimentary study.
Right. It must be sedimentation. So we have to, we have to make sure that we are analyzing sediment samples. We are trying to get samples or information from sedimentary deposits.
Right. And would you mind to explain a little bit about net ecosystem exchange, because that was mentioned with your Kaziranga thing, and how it is connected to this paleo study? Bitu, do you have
yes, that is also a very good question.
No, I was about to ask the same question, that we would like to hear something about the Kaziranga project as well.
Right. See, this Kaziranga project actually. So what we do? Firstly, let me just give you one our natural concept. See, what we think is that the human being is emitting huge amount of CO2. Now, where the CO2 is going? The extra CO2 added into the atmosphere, where they are going? And we all know that a significant part is going into the ocean, absorbed by ocean, and significant part also being absorbed by plants. Because plants do photosynthesis, they need carbon. So if you have some excess amount of carbon, they can absorb these things. So when you have more amount of carbon, it is expected that photosynthetic rate will increase and the plant's productivity will increase. So actually, this has been observed, and this is called this has been observed. The more CO2, actually, it is increasing the productivity. But productivity, of course, it cannot increase linearly. There will be a threshold. So I'm not going to that point. But what is happening is that our general idea is that forests are good absorber of carbon dioxide. So any forest, right? That is what we think. But now you have to, in order to test this hypothesis that every forest is a very good absorber of carbon dioxide, you cannot just assume until and unless you actually calculate how much carbon dioxide they are absorbing. So that is one of the best way to do. There are other methods are there, but eddy covariance provides you the very reliable method. So eddy covariance, what it does, basically you put some sensors on a tower, and above the canopy the sensor is placed, and then it measures very high frequency measurements of CO2 concentration and water vapor concentration. So if you measure high frequency means 10 hertz. So 10 at a 10 hertz rate, you are measuring CO2 and water vapor concentrations. So and when you measure these things, and after doing very lengthy calculations are required. So when you do these things, so you can calculate how much CO2 is being absorbed by plants and how much CO2 is being released by plants. So first is photosynthetic process and second is respiration process. But the measurements will not give you information of these two parameters. It will give you the net values. Supposing in say morning means when you have sufficient sunlight, so it will absorb more CO2 from the atmosphere and release relatively less amount of CO2 because of respiration process. But night times it gets just opposite. Night times there is no photosynthesis, so there will be no drawdown of photosynthesis from the atmosphere, so it will mainly release CO2. So if you take the absorption, if you take negative, means carbon is getting lost from the atmosphere. So you take it negative. So the total value or the net value, it will be negative during the daytime and it will be positive during the night time. So if you draw a curve, it will be something like inverse of bell-shaped curve. It will be something like bell-shaped curve with the tails are slightly positive, so they are representing the night time, and the other portion will have negative value, so it will have deep negative values. So now this is called NEE, net ecosystem exchange. Positive value during night time. This is you are doing on a daily scale. Let us say night 12 to next day night 12. So you are doing so this now the amplitude of this NE will tell you what is the capacity of a forest. Supposing and the unit is so many micromoles per unit time per unit area. So this is the unit. So now supposing say some forest will give you, let us say, value 20. So 20 micromoles per unit area per unit time is getting a negative value. You got the maximum NE is, let us say, minus 20. But another forest you get it is minus 40. For example, Betul in Madhya Pradesh, they have maximum value, it goes almost minus 40, minus 45. But in case of Kaziranga, we get maximum value maximum we get minus 20. So that means which forest is absorbing more carbon? So obviously it is Betul in Madhya Pradesh, that is Sal forest. But in Kaziranga, it is lower. So it is much lower. So now the question is why it is so? Why is Kaziranga is not a good so-called good sink of carbon dioxide? So that you have to answer. So if you can answer these things when you do very detailed analysis, you have means multi-year results, and then not only any, you have to calculate how much CO2 is being released from the soil. The soil actually soil there are a lot of organic carbon and they decay continuously. They are this bacteria, they are doing this decay process. So they are also continuously releasing CO2 from so you have to do soil CO2 measurements. You have to do, then you have to do some so-called vegetation property, means how much liter fall is there. That is, leaves are continuously shedding. So what is their contribution in transferring carbon? So they are transferring carbon from basically biosphere to basically soil. So when you do all the analysis, then you can have a very good idea about the so-called the carbon sequestration potential of a forest. So that is why we do this kind of studies.
So in Kaziranga, that the eddy covariance sensor that will measure the net exchange at the top of the canopy. So that
Yes.
in that measurement, the carbon emitted from the soil is also included.
Yes, actually, yes, it is included. It is, but it is included means it is giving you the net value of photosynthesis minus respiration. But you have to separately quantify. So you have to, the very simple process is, I mean, theory wise, you have to put a chamber. For example, you put a chamber, let us say you have a chamber, like dome shaped chamber is there. You put
Yes.
this chamber on the soil, and then you have a sensor. Or let us say you allow sometimes to accumulate air in the, because of bacterial process CO2 is getting emitted. So you have some mechanism, you can collect this air. Basically, it is full of CO2. So then you have a given time. So you calculate how much CO2 has been emitted. So this will give you, if you do these things, so it will give you the soil CO2 component, or how much CO2 is being released by the soil. So then once you have this information, and once you have any informations, then you can assess whether your NE is how much it is controlled by soil CO2 emission. Actually, the culprit what we have found in case of Kaziranga is that soil CO2 component is very high.
So is it, can it be due to rich bioorganism in the soil?
Yeah, actually bacterial, because basically bacterial decay. All these, there are a lot of huge bacteria. See, I tell you, probably all of you have experienced, supposing after a long time it rains. Let us say in generally March, April is dry time, but if it rains suddenly, you get some peculiar smell, right?
Yeah.
Yes.
That is, you know what?
That's carbon.
That's
that is
bacterial.
actually the carbon dioxide released by bacteria.
Okay, yeah. Oh, so the smell of the soil that we get after the first shower.
No.
Yeah, exactly. After the first shower. Yeah.
Oh, is it?
Yeah. So we get a smell after
It
a
is this
dry spell when it
after.
rains, like the first rain of monsoon, we smell something, right, from the soil.
Yeah.
That's basically the bacterial release of carbon.
Exactly.
Okay. That I didn't know that.
So the bacteria plays a very important role in carbon sequestration potential of any ecosystem.
Okay, so
no,
yeah.
I have yeah. Okay.
No, please go ahead.
Yeah, no, I just wanted to know, like, how to find a connection between the paleo study, because my question is very specific and very simple. I want to find a connection between the paleo study and then this NE. So how
Eddy
to
covariance.
yes
Yeah,
medical
actually
system energy. So exchange.
exactly.
How to put the
How to
connection
connect
between,
them. And
very
then
simple way.
it is a very good question. Now, for example, if we want to know, see, eddy coverage technique, it can give you, I mean, it can give you measured value. That is very accurate, but eddy coverage technique has been there only about India, maybe only about 10 years. 10, 12 years. And if you go abroad in Western countries, you have maybe 30 to 40 years. So before that, how the forest behave, so what kind of which forest has had more potential, which forest had less potential, all this information, we do not have these things. What happened in the, basically, if we want to say this carbon dynamic studies in basically carbon release in the past time scales, let us quickly one. Which one? Not my phone?
Yeah, I think your audio.
Yeah, a little breaking.
Is it my phone? Let me check.
I think this is fine now.
Is it okay?
Yeah, this is less, but
Okay.
oh, just a moment, sir. Just a moment. Yes, sorry.
My video camera got automatically switched off.
Yes, sir. Please go ahead.
See, I cannot. Now it's okay.
Yes.
I think somebody called me. That time video camera got disconnected. Deactivated. Anyway, so just to answer your question, so probably you understood what I said, that we do not have any information about what happened, this kind of, let us say, any, can we get any in the past time scale? That is a very simple question. Any we
Lost him again.
I think we lost. Can you hear us? Hello?
but I cannot see you here.
Oh,
Now it's okay.
yes.
Yeah. So, actually somebody is calling me. Yeah, so I did not accept these things. So if you want to know the any past time scale, let us say in few hundred years, so how to get it? So in fact, recently we have done this kind of work. So what we did here is that, now if you understand, probably you have some idea about treating process. Every year, this just like sedimentation process, trees also form annual rings. So now, in a given year, supposing the tree has sufficient nutrient and it has sufficient water, so it will have healthy growth. So ring width will be reasonable. But in a given year, supposing this tree did not get sufficient nutrient or sufficient water, so its width will be, its health will be, it will be slightly subdued. So what will happen? The ring width will be reduced. This is a very known fact. Means normal year, in deficient years, you have subdued growth. So it will be reflected in the ring width. So normal year, it will have higher ring width, and deficient year or drought years, drought condition, you will have lower width. So this is called the so-called dendrochronology, and IITM also it is done in dendro laboratory, it is there. So now we wanted to see, so what we have done, we have collected treating data, and we have collected this any data. Of course, measurement data is available only for 10-12 years. So we have taken some model data. Then we have correlated this model any data with treating data. Though the correlation is not very strong, but at least it gives you some positive correlation between these two. Means when you have higher growth, higher productivity, you have higher growth rate. So if you have this equation, if you calibrate these equations, then from treating width analysis, now supposing you are going, you are as a dendroclimatologist, so you collected a reading and you did this ring width analysis. Then using these calibration equations, you can get some idea about the past any conditions. So that is how they are connected.
Yeah, but what we will going to achieve as a climate scientist, what we are going to achieve climate information from this past any?
From these past studies.
Yes.
So
past any studies. Past any studies, past net ecosystem exchange studies, what climate information we are
what climatic information we'll get? For example, see any we have to know, for example, this when temperature is rising, as I told you, do the productivity of plants is increasing, or it is decreasing, or it is remaining the same? So actually in when, as I told you, if carbon dioxide increases, productivity is supposed to increase. This is called carbon fertilization effect. There is a technical term called carbon fertilization or CO2 fertilization effect. So more CO2 means the more photosynthesis. But now actually it is not a linear process. After it achieves a threshold value, the growth will be hampered, and in fact it may have negative effect on these things. With higher CO2, instead of giving you higher productivity, the productivity may go down. So did it happen in the past? Because past we know CO2 sometimes was very high. Sometimes, in fact, there are biological periods, sometimes CO2 was even up to triple what we have today. 3 times CO2 we had in the geological past. So the obvious question is, how can we get the productivity information in the past times? So particular time when temperature was high, so what was the productivity? So can we face or should we face the similar situations in the years to come, when we will have higher temperature, when we will have higher CO2? So what will happen to productivity? How it will, productivity, plant productivity is very much dependent, and we are very much dependent on plant productivity. Our food supply, plant is the primary food supply. So entire animal kingdom depends on productivity. So how productivity behaved in the past, if we get a very reliable information from the paleo studies, then we can apply this knowledge what may happen in the future.
So from these net ecosystem exchange information, we
Yes.
can very easily, not easily, but we can, in theory, we can get information about our condition or growth of agriculture or health of agriculture in the past, and then we can use that to project in the future. So
yes,
yeah,
similar,
as someone who works on like climate dynamics, I was wondering this net ecosystem exchange, from that information, can it give me some idea about circulation or climate dynamics? Is it in a way related, or is it the other way that my climate dynamics would impact carbon in some way, which would affect the agriculture and which would affect the health of agriculture?
Actually, see, with your question, it's a very, very nice question. But it's very difficult to say the cause and effect. But let me tell you, actually, they are very highly connected. It is like a chicken egg conundrum kind of thing.
Yes.
So, supposing you have circulation actually matters. So circulation, how but plants also play some role in the circulation. Particularly plants can produce a lot of vapor through transpiration.
Yeah.
And Northeast India is a place where huge amount of vapor is produced through circulation. So very recently, actually, probably you know that the Supriyo Chakraborty in IIT Bombay.
Rainfall recycling.
No, they have. I think they have recently submitted a manuscript. It is under review. It is not yet published. So what they are saying is that they are showing when the central India gets very heavy rainfall, Northeast India get subdued rainfall, and vice versa. And then that actually they are analyzing. Of course, it is not a new things. Many people have done, but they have done more, means
detailed,
more quantitative analysis. And actually this when the central India gets low rainfall and Northeast India, they get lot of rainfall through this evapotranspiration process, and that is mediated by plants. So when the plants are releasing huge amount of transpirations, then they will alter the dynamics, moisture dynamics. So they will create convection, and then the vertical circulation, particularly it will be affected. So they will in fact affect the circulation process in and even monsoon rainfall. So this way they are very much interconnected. So that will be,
let me stop it here, and we can have a I think longer discussion later. But before, because we are already 10 minutes past the due time, so if shortly we actually ask this question every speaker, each speaker until now, and we are trying to figure out like how to involve or how to motivate college and school students of Assam to pursue meteorology or climate science as a research career, and how to develop that environment even from the school. So do you have some small projects like for school students or college students so that they can
Yes, I think it's quite possible. It is very now. See, nowadays, means say I have seen this. Means now in our time, what we did not do now. Even the school children, it like slightly higher class, means class maybe 9, 11, 8, 9, 10, 11. They do a lot of project, and particularly the school students who are very serious, they can do in fact lot of work. Even simple, if you give this simple data. Simple climate data, and if you explain to them, they can do very good work.
Yeah.
And
Okay.
through this process, we can definitely motivate them.
Okay. Now I am asking, like, do you have something in mind so that they can like collect some samples or something like that, and they can experience? Or you don't have to answer if you like now, but if you have something in your mind, then that would be great.
The collection of samples. See, the collection. But one thing is, for example, rainwater. See, we do, as I told you, we do rainwater isotope analysis. If even school children, they can do this rainwater collections, but in regular basis, just a few days result will not give you any useful information. Supposing if you get 1 year, or if better, supposing 2 to 3 years, if you collect rainwater samples and then do the isotopic analysis, then you can produce isotopic records for few years, and that will be very useful for to understand hydrology and monsoon isotope meteorology, etc.
Okay. Okay.
Are there
Perfect. Okay.
just an addition, are there any easy proxies, like not exact measurements of isotopic analysis, but are there any easy proxies of doing isotopic analysis, or like any kind of analysis with the rainfall samples? Suppose one student collects samples of water for a year, or one school, under one school, a batch of students does it. Is it so that always you would need a sophisticated machine, or you can do it in some proxy way to get an average idea?
No, I understand your question.
Yes.
Means whether by doing some proxy analysis, whether we can get this information.
Yes.
So that is very difficult to say, because simply because no, they give very strong, they don't have very strong linkage with the isotopic values.
Okay.
So we cannot use this. Is the only way you have to do the isotopic analysis. For that, it is, only one way is to, after collection. Supposing if you want to, supposing you want to collaborate with people who have this, these sources to do
Facility.
isotopic analysis. So through this process only, you will you can generate isotopic records, and then that will be useful.
Okay. And how valuable will it be if somebody collects water in different parts of Northeast for 365 days?
If you ask this question, in fact, just now we are working on a manuscript. What we are trying to do is to, for example, see, you do the spatial distribution of rainwater, right? Means rainfall pattern. So, for example, in Western Ghats, you get very deep blue. Let us say deep blue means very heavy rainfall. But and also you get in Assam, it's very deep blue. Let us say. But now, what is the corresponding isotopic values? Generally, very heavy rainfall related to low isotopic values. So they are anti-correlated. So, for example, where you get deep blue means representing heavy rainfall, let us say you get opposite value of isotope. Let us say it is related to red. So now I want to do a spatial, I want to investigate rainfall. Many people have done, and now it is just like bread and butter. But there is no such paper so far exist for what is the spatial distribution of rain isotope in India. The only reason is that because data density, data network is very poor, extremely poor. And in Northeast India, probably we have only 4 or 5 places where it has been done. Entire Northeast area, hardly 4 or 5 places records are there. So that will not give you the reliable information about the spatial pattern of rainfall. So that's why it is the very first that we have to build a very strong network and do the isotopic analysis. Then only we can create a reliable map of isotopic map of rainfall in India.
Yeah. I think with that note, let's finish for today.
We can conclude, yes.
Yeah. So it was a very wonderful discussion, and I honestly didn't want to leave, but it's 15 minutes we are overdue. So, sir, thank you so much for this discussion. Yeah, and we will continue this discussion post for some time, 5 minutes or so. And before that, thank you everyone for attending our session, and thank you 𝗀𝗪𝗙 for this platform. Thank you Vidyut co-hosting, and I hope everyone enjoyed, at least we did. And we will meet you again in December with some other session. Until then, take care, and thanks once again. Bye.
Yeah.
Most welcome. You're most welcome. Thank
So,
you.
sir,